the stepwise regulatory pathway for manufacturing change and for creating a biosimilar are fundamentally the same. One has worked away in the background with only one problem over 2 decades. One has been very much in the public gaze, certainly for physicians like me, for a decade and has proved fit for purpose. Every approved biosimilar has been reapproved for every indication. You test a drug in the most sensitive indication for a biosimilar to pick up a difference. And yet its most important role might be in a different situation. So while filgrastim, you can test its sensitivity for difference in mobilizing white cells in normal human donors, how will we use it? I want to use it to prevent life-threatening infections in cancer patients going through chemotherapy. I want to see that that works. And that's an extrapolated indication. The first drugs often had requirements to do randomized trials in both situations, but increasingly we'll find that randomized trials aren't done for indications that we want to treat. So for example, for our colleagues in inflammatory diseases, they faced a class of drugs called tumor necrosis factor alpha inhibitors. These have been transforming the world of rheumatoid disease, of psoriasis, psoriatic arthritis, of the inflammatory bowel diseases, Crohn's disease and ulcerative colitis. Now, where a drug shares the same mechanism of action, you can extrapolate that it's the same from the most sensitive indication to say it's going to be the same in less sensitive indications, as long as the mechanism of action is the same. But you could be faced, for example, with discovering in Crohn's disease, are we certain that in Crohn's colitis and Crohn's disease that the mechanism of action is the same as in rheumatoid disease? Now, the regulators in Europe were convinced by the argument of sameness of the analytic and functional tests that notwithstanding minor variations in the chemical attributes of the drug, that functionally it would be the same. But not every regulator, for example, was so convinced. For our colleagues, perhaps watching this from Canada, they'll know that Health Canada were uncertain. They required separate studies and more time to look at the data for the extrapolated indication. And that uncertainty was echoed by our colleagues in Europe. In Britain, the British Society of Gastroenterology fired some warning shots about these drugs. They said, look, we understand the need for affordable care. We understand that these drugs have evolved over time. Infliximab, the first biosimilar in this class, had already undergone 35 manufacturing changes by the time the biosimilar came along. So variation had been controlled over time, but could they be certain it worked in extrapolated indications? And that was echoed by the European Crohn's and Colitis Organization. They said they had strong warnings to give about these drugs. Now, interestingly, after these drugs came out, many of our colleagues in Britain, we credit them with not only using the drugs, but recording the outcomes in big patient registries. And you can read one that came out in 2016, published by the Royal College, that showed in their post-authorization real-world study that there was no clinically meaningful differences. That's also been echoed by our colleagues across Europe, where many of these similar post-authorization database studies have been performed. And so not only has the British Society of Gastroenterology reversed its position statement, but so has the European one. And they now say from their experience of using drugs in extrapolated indications that again, the European biosimilars stepwise regulatory pathway appears fit for purpose. But for clinicians used to explaining the benefits of a treatment to a patient in terms of clinical trials, it's new, it's very uncertain. And so understanding extrapolation will be key for our confidence and our patients' confidence.